now. Just give people a chance to. Okay, guys, we'll get started in a few minutes. I'll just give people a chance just to get signed in. Now we'll get started now in the next minute or two. I just want to give people a chance to get signed in. So we'll get started in the next minute or two. Okay, hi there everyone. My name is Maeve Leahy. I am the head of advocacy and communications in the Irish Motor Neuron Disease Association, and I'm absolutely delighted that you could join us here this lunchtime. And we're thrilled that we have research expert Dr Roisin McMackin here to chat to us today. So Roisin is going to be speaking about how motor neuron disease affects activity in the brain. Now she's also going to discuss how the research is done, who can take part, and why it may improve medical care and testing of possible treatments moving forward. So she's going to give a quick overview of the research and I suppose recent results. Don't worry, there'll be plenty of time afterwards for questions and feedback. So just a little bit of housekeeping on that. If anybody has questions, you can either put your questions up into the chat function, and myself and my colleague Dervla will be keeping an eye on them throughout the session. You can also email them into info@IMNDA, and then once Roisin has finished with her presentation, we'll put the questions to Roisin. And then obviously after the session, if you still have further questions, please do email them. We'll send them on to Roisin and we can send around the answers with the link for the recording, because the session is being recorded and we'll be sharing it out via email and on our website next week. So, you know, keep an eye on that as well. So that's me done. So I'm just going to hand over to Roisin. Roisin, over to you.
Typically have to unmute myself.
Yeah.
Are you recording this now? Or
yes, yep, we're live. Yeah.
Good. It'd just be good to have the video. So thanks very much to Maeve and to Dervla and the IMNDA for giving me the chance to give this webinar. I guess I just wanted to have another chance. We do this every so often to just reach out and give a bit of an insight into the research we're doing and why we're doing it and what it involves. So I am an assistant professor in the discipline of physiology, and I still work closely with the academic unit of neurology in Trinity College. So my research is based around looking at how brain activity is affected in motor neuron disease and more specifically in amyotrophic lateral sclerosis. So just to give kind of an overview of who we are and who's doing this research. So I have a background in neuroscience and a PhD in clinical medicine. So I did my PhD with Professor Orla Hardiman, looking at this brain activity in ALS, and if we could identify medically useful measurements using this. And I'll go on to talk about that in a moment. And then I stayed with Orla Hardiman and the MND Research. So I've been working in ALS research for about 7 years now. And then, of course, our research is overseen by Professor Hardiman herself. So she's the professor and head of neurology in Trinity College, and she founded and is the head consultant at the clinic in Beaumont, as many of you will know. And then Dr. Nasser Al-Islami is an assistant professor in neurology as part of Professor Hardiman's group as well. And he has a PhD in biomedical engineering. So bringing kind of the maths and engineering background things. And then we also oversee an extensive team of PhD students and research assistants who have backgrounds in medicine, engineering, neuroscience, and psychology. So together we're quite a multi-dimensional team, trying to bring together all these different skill sets in order to tackle understanding ALS. I just wanted to give a very quick preamble, just in case there's any confusion, just to clarify that the studies I'm talking about today will be those which are involved in understanding MND to improve our chances of developing a successful therapy. So I won't be talking about what we call clinical trials, in the sense of what I'll be talking about doesn't involve kind of experimental treatments given to the participants. And the other thing, just as a side note, is I'm an academic doctor, not a medical doctor. So while I'm delighted to answer questions on MND in general and kind of the research and what we know about it, I just can't answer any questions about individual medical diagnoses or prognosis. You'll have to ask the team at the clinic or your own doctor about that. So I'm sure many of you are very familiar with what MND or ALS is, but just in case anybody is kind of new to the area, I just thought I'd give a quick overview of what is it in terms of what it does. So motor neuron disease is so called because it affects the motor neurons. So the motor neurons are the cells which your brain and your spine use to control your muscles. So they can be subdivided into the upper motor neurons. So those are the ones that connect your brain to your spinal cord. And then they pass the signal onto the lower motor neurons, which connect your spine to a specific muscle, so that you can send signals from your brain to your muscles to tell them what to do. So motor neuron disease is actually an umbrella term for lots of different diseases that affects the motor neurons. So a lot of people use MND and ALS interchangeably. Actually, ALS is the most common type of MND. So ALS, or amyotrophic lateral sclerosis, is medically defined by a disease that causes the disruption of both the upper and the lower motor neurons. Whereas we also have primary lateral sclerosis, which only affects the upper motor neurons in theory, which I'll get on to why I say that in a moment. And progressive muscular atrophy only affects the lower motor neurons in theory. And then Lou Gehrig's disease is what some Americans call ALS because of Lou Gehrig, who had ALS. So, as I said, the MNDs, the motor neuron diseases, are defined by the degeneration or the dysfunction of these motor neurons. But we know now that MND is not solely a motor neuron disease. So the nervous system, so the motor neurons and all the other signaling cells in your brain are these highly branched cells. So these neurons, I've just put a picture on the left-hand side of what one of these typical neurons would look like. They have these really tree-like branches. What those branches are for is so that they can take in a lot of information from other neurons and they can send out a lot of information to other neurons. So really these neurons, which do all the signaling that our brain and our spine, all the things that they can do, they don't really act on their own. They don't really generate any brain functions on their own. Instead, they act as part of these big networks of cells that are highly connected, and they're passing signals back and forth between each other in these specific patterns. And it's the route that those signals take and the patterns that those signals are sent in that is what generates the functions of our brain and our spinal cord. So actually, if you zoom out and look at the brain, it's made up of these really complicated and really multifaceted networks of these cells, which have these strands connecting them in different patterns. So ultimately, the whole brain is made up of one big network. But within that, we have different pathways or different kind of sub-networks that we know are responsible for specific brain functions. So obviously most pertinent to MND would be the networks that control movement. So I mentioned the upper and the lower motor neurons, and they connect your brain to your spine. But within your brain, there are lots of other areas that are important for movement. And I'm not going to get into all the nitty-gritty of that, but just to emphasize that it's not only the motor neurons that are involved in producing that movement. We have lots of other parts of the brain that are important for that. And then the motor neurons are located predominantly in this olive green area called the primary motor cortex. And they're the ones that then ultimately send the signal out of the brain down to the muscles. But we also know that there are all these other pathways in the brain that are associated with doing other things, like controlling our emotions, or changing what we're thinking about, or paying attention, or just thinking to ourselves. So there are all these kind of complicated networks involving different parts of the brain and the connections between them. So for a long while, you know, for about the last 100 years, it was starting to become clear that people with ALS did not necessarily only have movement symptoms. So it kind of became, it started to become reported in medical journals in the 70s, that some people with ALS would have other symptoms like problems with thinking or paying attention, or some things like that, which we would refer to as cognitive functions. But that was kind of ignored. And then some people said, well, it's not a true problem. It's a reflection of things like the movement problems causing difficulty with things like speech or communication. But since then, there's been a lot of research done and new tools developed so that we can more kind of objectively determine that that is the case, that ALS does not just affect those movement motor neurons. So, for example, MRI scan studies help us look at the structure of the brain, and we can see that some of the connections in the brain that are not to do with movement can be affected by ALS. Likewise, we can do studies looking at not the structure, but the function of the networks in the brain, and see that some of those networks are not functioning normally, and some of those networks are not the ones that are involved in movement. And this is reflected in the symptoms that some people experience. So approximately half of people with ALS will experience some form of what we call cognitive or behavioral symptoms. So things like, for example, difficulty paying attention, difficulty having the motivation to get up and carry out different plans and things like that. Not everybody, but some people are affected in this way. So all of this is, these images are all from different studies that we've done in Dublin to determine how different parts of the brain are affected by ALS, but they have all been backed up internationally. So this is kind of an internationally definitive sort of point of view that ALS is a network disorder. It affects networks in the brain and not just the motor neurons. So some people kind of ask, well, why are you so interested in these networks? I mean, clearly the major issue, the acute issue, is that the motor neurons are not working. So should we not just focus on that for now? Which is a good question. So the reason we need to look at both the movement and the non-movement networks in MND is there are several reasons. So, for example, as I mentioned, in the movement networks, it's not just the motor neurons which are related to the movement symptoms. So if we want to effectively treat the movement symptoms, it wouldn't really make sense to only focus on the motor neurons. We do need to consider the other areas that are connected to the motor neurons and what they're doing, because these are important targets for potential therapies. Also, Of course, we know that the non-movement symptoms like difficulty planning or paying attention affects people's quality of life. So, and not only the people living with MND, but also their carers. So we shouldn't just neglect this as kind of, you know, a secondary issue that we shouldn't pay attention to. Also, if we have a more detailed picture of what MND is doing to the brain overall, it can make it easier to diagnose it and to treat it. So as many of you will be aware, the diagnosis process in getting a diagnosis for MND is very slow and inefficient, in the sense that we don't have kind of one test that a doctor can do that says, yes, you do have it, or no, you don't have it. And that means that people can be misdiagnosed, they can be sent down the wrong pathway, they can undergo procedures that weren't necessary, and all of this delays the potential to get that person into a clinical trial when a drug might help. And then finally, we know that some people who have certain non-movement symptoms have a more severe experience of the disease. So the movement symptoms can progress more rapidly when certain other symptoms are present. So we would refer to how severe the disease experience is as the prognosis. And as you will probably all be aware, ALS affects different people extremely differently. So it's very hard to know how each individual will be affected. But by understanding not just the motor neurons, but the other aspects of how the brain is affected, we would be better able to determine how each individual will be affected. So in terms of why this is kind of medically useful, I just want to give a couple of examples of why being able to take these measurements of ALS would ultimately be beneficial for helping to treat ALS. So this is a very complicated diagram, and I don't want to go into it in too much detail or kind of give a science lecture about it, but it's I just want to highlight some key points with this diagram. So on the very left, we're looking at kind of when somebody is first conceived and developing. And then as we move towards the right, we move as the person is developing through adolescence to being an adult. And this is a good way of looking at how ALS develops. So we know that genes play an important part in ALS. In some people, it's much more of an effect than in others. So in some people, we can say, you know, having a certain gene or a couple of genes, certain mutations in those genes is the major contributor to why someone got ALS. In other people, it's more likely that kind of lots of very small effects from lots of different mutations in genes kind of increased someone's risk of getting ALS. And then as a person goes through their life, they will pick up experiences or exposures to different things in the environment, which will stack up on top of that environment, that genetic risk. So that ultimately that will bring the person closer and closer to a threshold for where they can develop ALS. So during this time, we know that way before the symptoms actually start, some of these factors are triggering the motor neurons starting to get sick and starting to malfunction. We don't exactly know where in the timeline this is, but we know it's substantially before the symptoms start. So during this time, you know, years probably before the symptoms start, the motor neurons are getting sick and we're losing a large proportion of those. Now, luckily, our brain is made in a way that we have a lot of reserves. So we can lose a certain amount without it causing a problem with our movement. And we can also use other strategies. So our brain can start kind of employing other networks that we wouldn't normally use to try and keep the functions going. And as a result of this, the motor neurons can be kind of quite sick before the symptoms start happening. So we're looking at the symptoms starting here, even though a lot of damage has already happened. And then even if the diagnosis happens quite soon afterwards, we're still quite late in the process. So then if we want to test a drug in ALS, we're really intervening with a drug very late when a lot of damage has already been done. So what we would ideally like is to shift the point at which we identify the disease back. So when there's less damage done, there's more healthy motor neurons, or at least motor neurons that are sick but are well enough to be rescued. And then if we could detect the disease earlier, we could get people into the clinical trials earlier, because the drugs are more likely to work if we can apply them when there's less damage to be counteracted. So we are hoping that by identifying things that we could measure in people with ALS, either very early on in when the symptoms start to appear, or ideally before the symptoms start to appear, that we could know who that we need to give these drugs to and start treating much earlier. So another reason why we need to understand, what is causing the prognosis or the difference in how people are affected in ALS, is because it's really important for how we test the drugs. So some people say to me, well, like, I wouldn't have wanted to know what was going to happen. It doesn't matter to me, why do we need to know what's going to happen to each individual person? Some other people would really like to have the certainty of knowing what is going to happen. And that's one reason why it's really important for us to get these measurements that help us predict how each person will be affected. But another reason is because the fact that ALS affects different people so differently is really affecting our ability to determine if a drug is working or not. So I'm going to use kind of a simplistic diagram to illustrate that. And I hope I don't kind of overly confuse anyone or undersimplify it either, but you can ask me afterwards if it's not clear. So this is a simplified example of, say, if we had 8 people that volunteered for a clinical trial and they all are living with ALS. So say if there was a way that we could predict how badly or how severely each person would be affected, imagine in this case, we would be able to see that 4 of the people were going to be more severely affected than the other 4. And I've put them in red and green respectively to illustrate that. Now, at the moment, we can't do that. So we just have to deal with the fact that we don't know, and we're just going to have to assume that they're all that all the people in the trial are going to be affected roughly similarly. So in order to do the drug trial, we need to put some people into what are called a placebo group, which means that they're not going to get an active treatment. They're just going to get something that looks and feels like the treatment, but it doesn't have the actual active ingredient in it. So we have the placebo group, and then we have the active drug group, who are going to get the active thing that we think is going to help in the therapy. And we just randomly assign people to these different groups. It's just a purely random process. But the thing is, if we had have known what the prognosis of these people was going to be, we would see that what we accidentally did is put all the people with the less severe prognosis in the placebo group and all the people with the more severe prognosis in the active drug group. Just as easily, we could have done the opposite, where we accidentally put all the people in green in the active drug group and all the people in red in the placebo group. But for now, let's say we accidentally put all the people who have a more severe prognosis in the active drug group. So most of the people in the active drug group were worse off to begin with. So we couldn't stop this because we have no way of predicting what's going to happen to the people in the drug trial. So now let's imagine that this drug works, that it has a benefit, and the people who have the quite severe disease end up having a less severe disease. So if we could see what their prognosis was going to be if we hadn't given them the drug, it would look like this. But now the people who had the more severe prognosis actually have a better prognosis. So we put them in yellow, so they're better off than they would have been. We don't know this because we don't have a way of measuring it. But what we will see now is, in a drug trial, you'll give the drug to the active group, you won't give the drug to the placebo group, and then you'll see who's better off. So because we don't know what the original prognosis of the people is, we just look at, who's doing better, or are the people who got the drug doing better than the people who didn't get the drug? But unfortunately, actually, in this case, even though the people who got the drug are better off than they would have been, they're still worse off than the placebo group, because while the drug made things a bit better, it didn't bring them up to the level that the placebo group were at the first place. So what the people doing the trial would conclude from this is that the drug didn't work because the people in the active drug group are worse than the people in the placebo group. So a drug that was helpful it looks like either it wasn't helpful, or it might even look like it made things worse. But this is all just because of that random chance that we accidentally put all the people with the worst prognosis into the active drug group. And so this is how genuinely valuable drugs could be lost, because we're not able to predict how individuals were going to be affected if they hadn't been given the drug. So say with this research or other research, we find a way to predict who's going to have a more severe prognosis than others. Then what we could do is take that measurement in all the people that volunteered for the trial and separate them into the groups like this. So now we have a more balanced set of prognoses across the two groups. Then we apply, so now we have these two matched groups, or sometimes it's referred to as stratified groups. Say we apply the same drug, and the same drug has the same effect. It makes the people who have the worst prognosis better. It brings them from a red to a yellow. The people in the placebo group who had the red prognosis, the bad prognosis, don't get any drug, so they stay in the red group. But the people in the active drug group are made better, so they go from red to yellow. Now, overall, if we compare the two groups, we can see that the people in the active drug group are better off than the people in the placebo group, and we have detected that the drug works, and that helps us to deem that the drug is effective, and we should approve it. So that's a very drawn out example, but I hope that illustrates why it's so important to be able to predict how different people will be affected. So then how does our research in Dublin help with all of this? So bringing back to the networks, if we are able to measure how ALS is affecting the brain networks, we are hoping that we'll be able to see the disease before it starts. So we're hoping we can measure that the changes in the function of these brain networks before we get to the point where this causes symptoms and ultimately leads to a diagnosis. So hopefully these measurements could help us predict that the disease is going to start and getting with a drug earlier. And we also are hoping that we can detect differences which are causing some people to have a more or less severe disease experience or prognosis. So how do we measure these brain networks? Sounds very invasive, but thankfully it's not. So we use two methods to do this. So one is called electroencephalography, or EEG. So this probably sounds familiar to some people because some people will have undergone what's called a needle EMG, which is where they measure the electrical activity in the muscles, but that is part of the diagnostic process for some people. And that is, I hear, very unpleasant and invasive, and that's not what we're doing. So just to clarify, we're not doing the needle EMG. So for this EEG, we place sensors on the head. So we have this cap and we just plug the sensors in. And they just sit on top of the scalp and record the brain activity, which passes from the brain. The signals pass through the scalp and the skull and can just be picked up by these sensors. So it's totally non-invasive. There's no needles, there's no blood, there's no electric shocks. It's just quite boring, frankly. We just have you do some simple tasks or just sit and let your mind wander while we record what the brain is doing. Luckily, it's very inexpensive, which means that it's very suitable for research, which unfortunately is very underfunded, but also would make it very readily applicable in a hospital situation where most countries are very underfunded in terms of their health systems. And in terms of participation, the participant just needs to relax in a chair. So there's no lying down, which I know is uncomfortable for some people, like in an MRI scanner. This is different. You just sit in the chair. And then we record those signals. So this allows us to measure all different networks all across the brain as the brain is doing different things. The other thing we do is something called transcranial magnetic stimulation, or TMS, which sounds very exciting It's not really. I mean, I find it interesting, but a lot of people, you know, in terms of participation, you just have to sit there and let your mind wander. So this, again, no needles, no blood. And the key thing is we're using magnetic stimulation here. So we're not using electrical stimulation. So the magnetic stimulation is not the same sensation as causing an electric shock on the head. So the magnetic pulses, some people don't really notice them. Some people say they feel like a tapping on the head. So we hold this coil over the person's head and deliver these magnetic pulses. And anybody who's into a bit of physics will know a bit how if you induce a magnetic field, that can then induce an electrical current. So our neurons work by electricity and in how they communicate. So if we just apply this magnetic pulse, that can induce the electrical current in the underlying neurons so that they start to signal between each other. So we do this by applying these pulses over where the upper motor neurons are, which is approximately here in your head. And if we apply them in a certain way, they will cause the upper motor neurons to activate and send a signal down through your spine to those lower motor neurons I mentioned, all the way to the muscle, and then the muscle will give like a small little twitch. And we just record those little twitches with a pad that we stick on the thumb. While the person relaxes and lets their mind wander and hopefully doesn't fall asleep. And by measuring how the muscle responds in response to different types of pulses, we get a very good measurement of how different parts of the movement brain networks are working. So some examples of what we've found so far, I mean, thankfully, we've actually made a huge amount of progress. I don't want to go into too much detail about the individual studies for people who haven't taken part yet, because it might bias how they take part in the study. But just to give some general overviews. So one thing we found is that we can show that we can detect problems in the non-movement networks before any non-movement symptoms appear, which is great. So again, going back to, we want to detect the problem as early as possible before enough damage has occurred to cause the symptoms. So we have shown that in the tests of things like attention, we can detect the problem in the attention network before it gets to the point of causing an attention problem, which would mean we could potentially test drugs on that before the symptoms have started. We've also shown that different people experience different patterns of brain network changes. So not everyone goes through the exact same process. So we now want to understand, does this require different medical intervention? What does this represent in terms of, are these different types of ALS? So we're still working on that. We've also shown that the early changes in the non-movement networks help us to predict how fast or slow the movement symptoms will occur, which brings me back to at the start of the talk when I said that we know that people with certain non-movement symptoms have more severe movement problems in the disease. So this is corresponding with that, where if we can detect these early non-movement network changes, we might be able to better predict how a person's going to be affected, which, as I mentioned, can help us with things like testing new drugs. We've also identified new information about how those brain networks are changing. So not just where they're affected, but how they're affected. And that will help us in refining what therapies we should develop. And finally, we've identified a number of new measures which are affected in ALS, or which are abnormal in ALS, and could help us improve how quickly we can diagnose ALS, but also improve how we test new drugs. And excitingly, we have a new a drug trial that will be starting. I'm not exactly sure when, but we're going to start using some of these measurements as extra measures to test if the drugs are working or not, where other measurements might not be good enough to detect if the drugs are working. So just to visualize this, on the top, this is a very kind of complicated figure, but this is a picture from one of our studies showing the four different types, the four different clusters of people with ALS and how different areas of the brain are affected in different ways. So as I mentioned, we're still working on understanding what's the medical implications of that. But interestingly, we found that these people with these different patterns of brain impairment, they stay they stay in their group, or they don't cross over these different groups. So these different groups don't represent different stages of the disease. They represent different patterns of the disease. So we want to look at how these different network types of ALS are medically relevant. And then on the bottom, this is just an illustration of, so this is a measurement, a brainwave that we record during one of the tasks we do in EEG. And on the left, you can see this red line has this negative peak, and that negative peak is missing in people with ALS. And what was interesting about this study is this difference was found even though the people with ALS did just as well on the task as the people who do not have ALS. So in illustrating that we can capture these problems before the symptoms are starting, and hopefully then intervene and have a better chance of treating the problem. So going forward, we still need to refine these measurements. So we need to do larger studies. We need to do replication studies in new groups of people with ALS to make sure we're confident in these measures. We're also looking at combining these measures to form a better picture of how each individual person with ALS is affected by ALS in terms of what areas of the brain are affected and how this relates to their prognosis. And as I mentioned, we're introducing some of these measures into a new drug trial in Ireland to see if these measurements can help us better detect which drugs are working and if they're acting on these abnormal brain networks to help counteract the problem. So the research is always ongoing, so we always need more participants, actually people with ALS, but also people who do not have ALS as what we call control participants would also be welcome. But anyone over the age of 18 years old is welcome. The study takes part in St. James's Hospital, just near Heuston Station in Dublin, just off the Lewis line. And if you'd like to take part, please do feel free to contact me at my email address here, or give me a call or a text or a WhatsApp on this number, or just contact the IMNDA and they'll be happy to pass on some information. But if you do get in touch, you're not signing up immediately. We'll just give you more information. And then if you decide you don't want to do it, that's also no problem. So that's me. So I think I will pass back over to Maeve to see if we have any questions.
No, I have a few, all right, Roisin. That was excellent, by the way. Thank you so much. It was so informative. And as I said at the beginning, this has been recorded, so I'll have the link. We'll send it around to everybody who registered, and we'll also put it up on our website, up on the community hub, and we'll get a little bit of word out there on social media, because I have to say it was absolutely fascinating. Now, I have a few questions that have come through. Now, Andy emailed, and he would just be grateful if you could clarify for him whether motor neuron disease is a brain disease, a brain abnormality, or a motor neuron disease, or a combination. He has had the disease for over 45 years, and in his opinion, it's a motor neuron disease. But he would just like some clarification on that.
Yeah, that's a great question. And I think it's a combination of two things. So it is a motor neuron disease. Definitely the kind of uniting characteristic of everyone with ALS is that they have problems with their upper and their lower motor neurons. But as I kind of talked about in the talk, these motor neurons don't exist in isolation. They have all these connections to other neurons. So they can't sort of be impacted without the cells that are connected to them also being impacted. So it's definitely primarily a motor neuron disease, but it does affect the brain more widely.
Okay, super. I think that definitely answers this question. And now, and actually just, I suppose, touching on some of the subjects that you closed with around the participation in studies in St. James's, we have another question in from Dennis. And Dennis said he participated in an MND study being carried out by Professor Hardiman, and it was between St. James's and Beaumont in 2017, 2018, 2019, and it was organised by Dr. Owen Finnegan. And he is wondering if part of this presentation was informed by that research. And unfortunately, due to COVID, the study was actually stopped. But he was wondering, was there a plan to commence the study? Now, I'm aware you mightn't have the answer to this, but I look at Dennis, we'll try to get an answer for you, be it now or after.
Yeah, so certainly. So Dr. Finnegan, he was doing his PhD at the same time that I was doing my PhD, and his research, which would have been the study Dennis took part in, was focusing on MRI, so looking at the brain structure. And then I kind of do a complementary study where I look at the brain function. And so some people might have done a day where they came in and did an MRI and an EEG, or did both on different days. So Owen thankfully passed his PhD, and he had some great findings from that research and has published them. So certainly, if the person is looking, if Dennis is looking to see the results of that study, some of them have been published in journals already. And Owen now has unfortunately left us from the research side of things to pursue his medical career. So he is still but our MRI research is still ongoing. It's led by Ranga Chapika and overseen by Dr. Peter Bede. So we still do that research.
Okay, perfect. And look at, maybe that's another research webinar that we can do later on, Dennis. So don't worry, we'll get some more information back to you. And again, just I suppose we had another question in from Roy, and you know, what I could see at the end of that was things are moving in the right direction with your new measurements, and that there will be more clinical drug trials. But he's wondering, are we likely to see any new drugs in the near future?
Yeah, so I mean, when I started working with ALS 7 years ago, there was one drug, riluzole, and that is still the main drug that's used. It's been shown, unfortunately, it's not as good as we would like, but it has been shown numerous times that it definitely does work. Since, in the last, I say 3 or 4 years, there's been 4 different drugs, I think, that have come to the forefront. One that's particularly for a genetic type for people with a specific gene called SOD1, which is not very prevalent in Ireland. But the fact that therapy is so effective is very promising for the other genes and people who have other genes that cause ALS. There are some other drugs. One of them is developed by a company called Amylyx. And there's a big push in America to get that approved. I think it has been approved recently in America. The key thing is that what the European standards for what is a good drug and the American standards and the Japanese standards are different. And I think actually the European standards are good. I mean, I totally understand that people are like, you know, give me anything that you think is going to work. But unfortunately, you know, there are some drugs that are licensed in other countries that it's quite clear that they're not effective and they cost a huge amount of money. And so I'm quite glad that some of them are not licensed here in Europe. But in terms of good drugs, definitely, I think the Amylyx AMX0035 is probably going to be approved in Ireland soon. There's another one called Adaravone that's licensed in the US, and they're looking at making that into a tablet, because at the moment it's only available as like an IV drip. And the evidence is questionable, but they're looking at making that into an oral drug, and hopefully, if that's successful, it would be made available in Europe. So, I mean, considering there was one drug and now there's about 4 drugs on the cards, that's huge progress. And I've seen a lot of promising results from early phase trials. I would just always warn a bit of caution around, sometimes with the media, these early phase trials find these results that look great, but then they don't really carry through when they're tested in a bigger study. So just always be a bit cautious of, sometimes the media likes to overhype things, but I am very optimistic that there is progress being made.
Brilliant. And if there is any information on early phase trials or things that people have seen, please do send them in to us. And as Roisin touched on, we can find out the answers for you, or if we don't know, we can forward it on to Roisin and the team, and then get that information out across to you. Now, I have a few more. I have another question from Tamara, and she said, she's taken part in research which is still ongoing, but for people who are taking part in research, is it possible to get their results? I don't know.
Yeah, so We're obviously more delighted to, engage with anyone who wants more information. The key thing I'd have to say is that, by all means, we can provide you with information on what we measured. We can give you the actual data. We can give you pictures of what we've measured. What we can't do is say, this measurement means this is going to happen or that's going to happen, because that's what we're kind of doing the research for, is to determine what those measurements mean. So I would be more than happy to say to somebody, this is what your data looks like, and this is why we're measuring it and what we're measuring. But I can't say, if you have a value of below 2, that means X, and if it's above 2, that means Y, because we haven't, it's not clinically defined like that yet. Is that, if you get me?
Yeah, and I think that does go to the heart of her question. I have another question here from Sandra. You mentioned that cells beyond rescue, so that there was cells beyond rescue. Can cells ever repair with the right drug?
So, yeah, I think that's an excellent question. And it really goes to the heart of why I'm particularly interested in looking at brain function. So imagine you have these neurons and they're living and they're happy, and then something happens, genetics and environment, and they're not happy, they start to get sick. This is the ideal point where we want to get in with the treatment while they're still alive and they're still doing their job. They're just not doing their job very well. When we can kind of hopefully halt what's happening and rescue them. What is suboptimal is if, for example, on some more structural studies, they can see that large areas of the tissue in more severe late stage disease have died. Okay. At that stage, it's very difficult to bring back more cells, because neurons they do in certain circumstances divide. But say, for example, like if you cut your skin is really good at making new cells and repairing and dividing, whereas your neurons are pretty bad at that. Okay. For about the last 5 or 10 years, it's become clear that some neurons do divide and there is some repair, but it's definitely not as good as things like our skin or our hair. So it's not undoable, but certainly there's been, really awful, some companies touting, stem cells to regenerate new cells. And there really is very poor evidence of that. And those treatments are actually quite dangerous. So definitely, ideally, what we want to do is get in with a treatment when the cells are still alive. They're just a bit unhappy.
Right, of course. Now, Aileen would like to know, if you were to detect brain deterioration before symptoms are apparent, wouldn't that mean screening would need to be available?
Yes, that is true. So at the moment, what we'd be looking at is, for example, if there was a family where multiple people in the family, more than we would expect to normally have it, would have ALS, then what we would maybe look at is there other people in the family we can test or screen, as you said, to look for evidence that this is happening. So that would be where we'd apply that. I guess the difficulty, as I said, is knowing who you would screen, and that would probably be mostly based on if other people had quite a high prevalence of it in their family, or we had some other reason to suspect it. But that, it is a difficult question. And I guess the other thing is coming back to, the health service. If, there's only a certain amount of money, unfortunately, in health, in hospitals, so we couldn't just kind of screen everyone, certainly at the moment. But so that's where we'd be looking at going in terms of the pre-symptomatic, at least in the near future. But we don't know about kind of further out.
Okay, perfect. I think I might have one more here. And it is kind of following on from that theme of testing and screening. Pardon me, sorry guys. Sarah would like to know, her mum has motor neuron disease, and is there a gene that me and my siblings can be tested for to see if we have the same gene?
Okay, yeah, so this is it's a little bit complicated. So if her mother is known to have a genetic type, so people with ALS can be screened for certain genes if it's suspected that there's a genetic component, and if that person, if the person with ALS is found to have a gene, it may be possible that other people in the family could get tested. But I'm not, in terms of how the health service works, really the person would have to talk to Professor Hardman, because genetic testing is extremely expensive. So it's not sort of, I don't think it's typically offered unless, for example, there are lots of people within one family, because there are also people, you know, single people within a family, and even if they do have a genetic kind of underpinning, no one else in their family ever gets it. So it's not to say that if a relative has a known gene associated with it, that others in the family are going to get it. And then the other thing is that the genes that we know drive motor neuron disease, they're not, this is to use the scientific term, they're not fully penetrant, which means that you can carry the gene and not get the disease. It just means that with the gene, there's a higher risk. Some people say it's because it brings you closer to the threshold, so that you need less kind of to encounter less environmental factors to then get the disease. So I guess the two things I would say is talk to Professor Hardman if you're concerned about genetics, but only about 10 to 15% of cases of ALS are what we call familial, where multiple people in the family get it. And the second thing I'd say is it's not a case of if you have the gene, you're definitely going to get ALS. So don't, especially if you get some sort of genetic test done and you're worried about the results, it's not a, you know, if you have the gene, you're going to get it kind of thing. Okay.
And you know, Sarah, if you want any further information, please email info@imnda.ie and we can forward you on more information. And as you're answering all these questions, Roisin, I'm like, God, there's several webinar topics popping up. So if there is something that people want to hear more about, please do let us know. Again, email info@imnda.ie and we can look at getting these topics covered in the next few webinars that will be coming up over the next few months. Now, the questions just keep coming. So I have another one here from Teresa, and she's just talking about participating in research. And she's like, participating in a trial of research, having lost my mum to MND, is such research trial not suitable for someone who has had a family member with MND?
That's a great question as well. So what we used to do is exclude people who were first or second degree relatives of people with ALS from the studies, not because there was any kind of substantial evidence that they you know, there was anything different about those people, but just because in terms of the scientific community, it was always seen as kind of just better to be sure that you had, there was no potential kind of muddying of the waters by including people with ALS or genetic relatives of people with ALS. But that's very unfortunate, because a lot of people who are relatives of people with ALS understandably want to take part in the research. So now what we do is we welcome people who are relatives, genetic relatives of people with ALS to do the study. We just ask that they take part as relatives of people with ALS as opposed to controls, just so on the very off chance that there is something different in the relatives, doesn't mean they're going to get ALS, but you know, if there is something different about that group, that we can detect that separately from people who don't have any genetic relation to people with ALS. But yes, it is also extremely valuable if relatives of people with ALS take part. So yeah, certainly we have research ongoing that those people could take part in.
And if you're interested, again, get in touch with Roisin on the contact details or get in touch with ourselves. Now I have another one. This is from Aileen, and she said, I have been diagnosed with PLS, which you have said involves the upper neurons only. But you said theoretically, what did you mean by that?
Yeah, so the classic diagnosis of PLS, PMA and ALS has classically been that people with PLS only have upper motor neuron degeneration, people with PLS only have lower, or PLS only have upper, PMA only have lower, and ALS have both. What I mean by in theory is that so typically people with PLS don't go on to develop. Remember I said there are upper motor neuron symptoms and lower motor neuron symptoms. So typically people with PLS only show the upper motor neuron symptoms, things like hyperreflexia or weakness or things like that. But as I said, those neurons don't exist in isolation. They are connected to other neurons. So I just mean by in theory that just because it's upper motor neuron only doesn't mean that the cells connected to those upper motor neurons might not also be part of the disease. I'm not trying to kind of scare anyone or freak anyone out, but I just mean in terms of if we wanted to better understand PLS, we should not just focus on the upper motor neurons, we should also focus on the cells connected to them to see if that could help us understand what causes PLS and, look for therapeutic targets.
Well, listen, Roisin, I just have to say you've been fantastic. It's been an absolutely brilliant session. And as we were saying, hopefully we'll do more of them. I think that's all from the questions so far. But if anyone does still have any remaining questions, I urge you, send them into info@IMNDA. We'll fire them across to Roisin, or we'll send them around the houses to try and get them answered. We will be sending out the recording link to everybody registered next week. And as I said, it'll be posted on the website and other channels. Roisin, thank you so much. We're going to leave it there and we'll let everyone grab a cup of tea and get their lunch. But listen, we really appreciate it. Thank you so much.
Thanks a million. Thanks, everyone.